The 1950s and early 1960s were a time of optimism. Advances in medicine promised better health and longer lives. But not all medical breakthroughs lived up to their promise. This is the story of one such drug, thalidomide, also marketed as Contergan. In 1957, the German pharmaceutical company Chemie Grunenthal introduced thalidomide as a sedative and treatment for morning sickness in pregnant women. It was hailed as a wonder drug for its effectiveness and lack of obvious side effects. However, in the following years reports of babies born with severe deformities began to surface, including the shortening or complete absence of arms and legs, as well as malformations of hands and feet. It is estimated that 10,000 infants were affected worldwide, still excluding miscarriages. So the hunt for the cause began. In December 1960, the Scottish physician Leslie Florence firstly raised suspicions on neurotoxic effects caused by thalidomide. Initially, the company Grunenthal denied the connections between thalidomide and the adverse effects, but due to growing pressure were forced to request a prescription-only status in May 1961. Dr. Widukind Lenz, a German pediatrician, was among the first to notice a pattern of severe birth defects in babies born to mothers who had taken thalidomide. In late 1961, he began to document cases of phocomelia, the medicinal term for malformed limbs, and other congenital anomalies. Around the same time, Dr. William McBride, an Australian obstetrician, observed similar birth defects in his patients. In December 1961, he published a letter in The Lancet noting the correlation between thalidomide use and congenital abnormalities. Following an anonymous letter, published in a newspaper article on November 26, 1961, Grunenthal finally withdrew Contergan from the market the next day. But why was thalidomide approved in the first place? And how could these drastic effects be overseen? It is important to note that knowledge in the 1950s and 1960s about the safety of pharmaceuticals was not as far advanced as it is today. Worldwide there were no strict guidelines for developing, producing, or marketing pharmaceuticals as there are today. Only after the thalidomide tragedy, procedures for authorizing and monitoring medicines that we know today were established. First of all, the understanding of teratogenicity was insufficient. Teratogenicity describes the ability of a substance to cause developmental abnormalities in embryos or fetus. In fact, it has been argued that, prior to the thalidomide tragedy, the general belief was that the placenta served as a barrier to protect the fetus from exposure to any agent. Although at that time there were already some indications linking alcohol to negatively affect a fetus. Furthermore, even though animal studies were conducted pre-market approval, these failed to reveal significant teratogenic effects. These studies included, among others, rats, rabbits, guinea pigs and swine. In addition to this, the drug was not tested on pregnant animals before approval. At that time, the necessity for teratogenic testing on pregnant animals was not fully recognized or mandated. It is clear that animals are different to humans and each animal has different tissue for distinct functions. That means that specific studies for investigating different aspects like teratogenicity, neurotoxicity or absorption and distribution of drugs have to be conducted using different animals. Using animal studies purposefully only evolved over time with an increase in knowledge. Also, no long-term or sufficient clinical trials were performed. The initial clinical trials in humans focused primarily on the drug's sedative effects and its efficacy in treating nausea and insomnia. These trials did not include pregnant women, and no long-term follow-up was conducted to monitor potential birth defects. Side effects were unfortunately recognized too late. It took several years for the connection between thalidomide and the severe birth defects to be established. The diffuse and delayed nature of the adverse effects made it challenging to link them directly to the drug. Had there been a strong focus on the safety of thalidomide instead of only investigating the efficacy, this tragedy may have been avoidable. Early evidence of neurotoxic effects should have been a sign of caution. The thalidomide tragedy marked a drastic event in the history of pharmaceutical development, leading to profound changes in drug approval processes worldwide, emphasizing the importance of rigorous testing and regulatory oversight to ensure drug safety. Since the thalidomide scandal, drug approval processes were drastically changed worldwide. Drugs have to go through strict safety, efficacy and quality requirements before approval. This is ensured by a number of different steps. The approval processes are extremely coordinated and knowledge exchange between pharmaceutical company and administration offices are rigorous. Furthermore, the administration agencies cooperate with each other, for example the Food and Drug Administration in the US and the European Medicines Agency. Uniform international standards have been developed to maintain quality in development, especially clinical trials and later production. All requirements of major administration agencies refer to guidelines like Good Manufacturing Practice (GMP), Good Clinical Practice (GCP) and the ICH guidelines determined by the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. A strong focus on strengthening the requirements for preclinical and clinical testing of teratogenic effects was also established. The importance of pharmacological safety studies, including reproductive and developmental toxicity studies, was emphasized. Another highly important step to evaluate drug safety is the pharmacovigilance system. This is a system for monitoring drug safety post-approval and ensures detailed post-marketing surveillance of new drugs. If adverse effects would come up, these could be evaluated quickly and efficiently to take immediate action. It is highly likely that no one could have anticipated such a clinical tragedy associated with thalidomide nearly 4 decades ago. The thalidomide incident directly led to significant reforms in drug development by exposing numerous deficiencies in the approval process. In response, new global regulations were implemented, introducing strict regulations, restructuring organizations like the FDA and European Medicines Agency, and enhancing patient and consumer protection through a more rigorous informed consent process. Today, manufacturers are required to demonstrate the quality, efficacy, and safety of a new drug before it is authorized to be tested on humans. Additionally, all drugs must now undergo formal testing for teratogenic effects, leading to rapid advancements in the field of teratology. Aside from the social and legal implications, these scientific advancements have provided valuable lessons. As we continue to seek out promising therapies, it is crucial that we maintain a balance between research progress and prioritizing human safety. Progress is a process. However, that does not mean that mistakes are inevitable, and serious ones, like in this case, should never have happened. The error here concerns several levels: irresponsible approval, aggressive marketing and careless prescribing, among other things. Especially in the field of human health, the goal must be to achieve maximum progress with minimal errors. Pharmaceutical development now has very high standards. Medicines must undergo lengthy processes to demonstrate quality, safety, and efficacy before they can be approved for human use. These standards are based on research findings and are constantly updated with new discoveries.